Alright, so we're going to start to try something new, and then hopefully you guys can bear with me. As you remember, we were talking about the different types of chromosomal aberrations, and so we talked about losses of chromosomes, monosomies, and we've talked about duplicated chromosomes, or ones that you receive 3 or more copies. So this would be your polyploid situation, where you have more than 2 copies. And so our example there was Down syndrome. So our next topic is going to be polyploidy. Now, if you remember what we talked about so we said monosomies was loss, and diploidy or triploidy, these are examples. Diploidy is the normal, but triploidy would be an example of a polyploid. And as we've said before, polyploidy is actually more common in plants than it is, or can be tolerated better in plants than it can be in mammals. So polyploid just simply means that we have more than 2 haploid sets of chromosomes. The naming of a polyploid is then based on the number of sets of chromosomes that you have in there. So we talked about a triploid situation with Down syndrome, so that was a trisomy 21, so it was just 3 bodies. Now we're talking about sets of entire chromosomes. So this is a triploid individual, or a 3N individual has 3 full sets of chromosomes. So if 2N has 2 full sets of chromosomes, then their 3N will have 3 sets of chromosomes. In the same idea, a tetraploid would have 4 complete sets of chromosomes, so they would be considered 4N. And pentaploid organisms actually have 5N sets of chromosomes. So this would be 5 complete sets of chromosomes. And yes, you can keep on going and going and going. Polyploidy can originate a couple of different ways. So you can have the addition of 1 or more sets of chromosomes that are identical to the haploid complement of the same species. If you remember our chart previously where we were defining these different terms, this is autopolyploidy or self-polyploidy, so that you have 5 or 4 or 3 complete sets of chromosomes from the same individual, so from the same species. Or you can actually have a combination of chromosome sets from different species that are a consequence of interspecific matings. This is allopolyploidy. Okay? So in this sense, you have different species that are interbreeding, and so you have different sets of chromosomes that are actually available. So this is an example of autopolyploidy and allopolyploidy. So we're going to look at autopolyploidy first, hence the red outline here. So this is where we're going to start. So we're going to look at autopolyploidy. So remember that each additional set of chromosomes is identical to the parent species. So what we're looking at here is this is our normal complement of chromosomes. Okay, so this is going to be your 2N, meaning that we have 2 copies of each chromosome. Okay, so this is going to be our diploid set. So this means that our parent here has 3 chromosomes, so this is our complement, and then we have 2 copies of each chromosome. Okay, in order to achieve a triploid, okay, so this particular complement here, we're going to have 3 copies of each chromosome. Okay, so basically what this is you're looking from the diploid complement above to the triploid complement below. And so you've gone from 2 normal sets to 3 normal sets. So each additional set of chromosome is identical to the parent species. So if we are going to say that 2N is this complement, okay, then we're going to say that this triploid situation is actually going to be this situation. Okay, so that we have a triploid complement there. Now if you'll notice next door, you also have a situation where you have a tetraploid. So this is identical to what we have in the triploid set. We just have here, we have 4 copies of each chromosome. So this would be our tetraploid, or A complement. Okay? So autopolyploid individuals are typically larger than diploid forms. If you have to think about it, you've got all this other DNA that you have to stash somewhere, so this has got to be present in the individual. So usually what happens is you're not producing more cells, you have larger cells. And so in this sense, you have to have a larger size nucleus to hold all the DNA, so your cells are larger. That's going to mean that your plants are usually larger. And so most of your horticultural or commercial plants that are actually sold out there are typically polyploids because of this. Let me see if I can extend the screen up. I'm not sure that that's working. So down below here there is a statement that says plants are usually more valuable if they are polyploids because there is a horticultural or a commercial value placed on these types of plants because they are bigger plants. So let's now look at the oh, it went backwards. Look at that. So now let's look at allopolyploidy. So allopolyploidy, different, okay? So if we're starting out with an allopolyploid, notice that we have an X here, okay? So that means that we're going to be crossing these 2 individuals. Both of these individuals are starting out as diploid, okay? So this means that this is a complete complement for this individual and this is a complete complement for this individual. So if you then combine these 2, you're actually, by nature, are going to create a tetraploid because we now have 2 complete sets of chromosomes for each of the different species that are represented. So each additional set of chromosomes is different from the parent species, and this happens when you have a haploid ovum that's fertilized by sperm from another species. So we have a 1N equals A that's fertilized by another species, 1NB. We now have an AB hybrid, okay? But what happens is, in order to make these something that you can actually divide and produce gametes, there is a natural or induced doubling of the chromosomes. So we go from a haploid situation to a chromosome doubling so that you have your tetraploid. So this is going to be your tetraploid. So this is going to be A, and this is going to be B. So this is going to be your tetraploid situation where you have an allopolyploid and you have 2 sets of chromosomes from each species. Now, as you might imagine, this cannot happen between lots of species because you probably need to be closely related for you to be able to actually utilize this particular form so that you can produce individuals. Otherwise, hybridization is probably not going to be successful between 2 very different species. So an allotetraploid is going to actually come from, typically, hybridization of 2 closely related species. Now, if you'll notice, it says here that your AB individual, so the one, your haploid complement A and your haploid complement B, and remember that A comes from species 1 and B comes from species 2, produces a sterile hybrid. So what this means is that every individual that's going to be produced, any gamete produced from this individual, will not be able to reproduce. So you can't produce a gamete that has a complement that the other gamete can actually recognize. Now, if you undergo a doubling process and produce an AABB individual, this is now considered a fertile allotetraploid because you can have an equal distribution of the chromosomes in this particular situation, and you would have an unequal distribution of chromosomes if you have this individual producing egg or sperm. So if both of our original species are known, the term amphidiploid is preferred. So if we know the name of the species, so if we say that we understand that this species or this parent is a grass species, and it is panicum, and we know that BB is, actually let's do this. Is also a grass species. Okay, but we know that this particular species is not panicum, but it is a different species. Okay, so if it's Scirpus, so if we know that this particular, that we have AA and we have BB, and we can actually determine where they come from, then we actually prefer the term amphidiploid. So this is a diagram of how we actually produce either a sterile hybrid or a fertile amphidiploid. Remember that amphidiploid means that we know which species our chromosomes are coming from. Okay, so let's look at this diagram. This is one of those where you guys turn your brains off, but let's look at this. So we know that we have AA and BB. So this is exactly where we're starting from the previous slide. Okay, so notice that this is a diploid individual. So this is one complete chromosome set here, and this is the second complete chromosome set. Again, here, this is a diploid individual because we know that we have one complete chromosome set here and one complete chromosome set here. So you have 1 and 1, 2 and 2, and 3 and 3 of our species 1. So A1, A2, A3, and we have 2 copies of each. And from species 2, we have B1 and B2. These are complete sets of chromosomes, and we have 2 copies of each. So that's a complete set. So we're 2 in. Now, if this particular species actually produces a gamete, the haploid number is going to be 1 of each, correct? So then if that's the case, if you then produce a haploid number here, you're only going to have 1 of B1 and 1 of B2. Okay? So if these 2 gametes are actually fertilized, so if we undergo this process and we have fertilization that's actually occurring between these 2, then we're actually producing an individual that has a complete chromosome set from A and a complete chromosome set from B. Okay? Now, this individual is considered sterile. Here's the problem. How many chromosomes do you have? We have 5 chromosomes. So how do we divide 5 chromosomes evenly to then produce a gamete that's going to have an equal distribution from this particular individual? So remember that this individual is the parent, so this is the adult that will hopefully, or if it can, produce gametes. But the gamete that this is going to be produced is going to be unequal. Unequal chromosomes. Okay? Now, if you actually have a chromosomal doubling, and I'm going to erase some of this stuff so that you can keep up with, it doesn't become too messy, because I think it's pretty messy right now. But so if we have a chromosome doubling, okay, what happens is as you move from from this particular individual, which is a sterile hybrid individual. Okay? If we have a chromosomal doubling that actually occurs in this particular phase, we will actually end up with 2 copies of chromosome A1, 2 copies of chromosome A2, and then 2 copies of chromosome A3. Okay? But notice at the same time that we also now have 2 copies of chromosome B1 and 2 copies of chromosome B2. Okay? So now what happened here is that we have restored an equal number . So we've restored an equal number of chromosomes. And we now have a diploid number of each chromosome set. Remember that A chromosome set is from one species and B chromosome set is from a second species. Now what can happen is that you can actually form equal gametes. Okay? So notice that this one is now A, B, and this one is now A, B, because we have a haploid complement of A and we have a haploid complement of B in each of those gametes. So now if these 2 gametes were to meet with each other, you could actually end up then producing back to a fertile amphidiploid. So in this sense, if you're thinking about this from a horticultural standpoint and you want to produce individuals that only you can sell, you're probably more interested in this particular individual here. If you are looking to sell a plant every single year, if you have an annual. If you are a person who wants to not have to buy a plant every single year, then you're probably interested in this particular option. Okay? And so in that sense, you could actually have 2 different objectives here. But fertile amphidiploids actually do occur sometimes in nature, and there are sometimes that it happens naturally. We don't really understand the mechanism of what would trigger it to actually produce doubling sets of chromosomes, but we do know that it does occur in nature, and it may be a response for the fact that that plant cannot reproduce. However, you can also do this, so if you are a breeder or if you're somebody who is interested in producing a particular variety for sale, if you will, then you can actually produce this as a result of somatic cell hybridization. I'm not sure what happened there. Nope. Here we go. Sorry about that. Learning new things. Amphidiploid plants can be produced by somatic cell hybridization. So what happens under this particular one is you can actually remove a cell from a developing leaf. You can treat it, which actually basically what you're doing is you're removing the cell wall. And now that you've removed this cell wall, basically what you have, all of the cytoplasm has left, and so what you have is a protoplast, which is basically a nucleus with a little bit of cytoplasm that still surrounds it. Then you can take these protoplasts and then you can actually maintain them in a culture. And what I do mean by this is something that you would equate to something you have in a microbiology lab. So you can actually maintain it in a culture and induce them to fuse with each other. So what that means is you are inducing the protoplasts to actually fuse together and produce somatic cell hybrids. Okay? So in this sense, what you can do is you can actually produce, what was that other term? Do we remember what it means to, or how the term changes if you know what 2 species they are, and if you are actually inducing 2 protoplasts to fuse by themselves, I'm hoping that you know what species they came from. So in this case, we would actually be creating an amphidiploid, right? So an amphidiploid plant that is produced by maintaining 2 particular protoplasts that you know what species they came from, and you can produce a somatic cell hybrid. All right, so we will actually look at this in class again. I want to make sure that you understand what's going on when we have allopolyploidy and when we have autopolyploidy, so that you can understand what the process is. So, I'm not sure. Okay, so endopolyploidy is basically what happens when we have only certain cells of an otherwise diploid organism that are polyploids. Now, this does happen in nature, and it happens with vertebrates. So in the vertebrates, we have liver cells that are either 4N, 8N, or 16N. We don't really understand why endopolyploidy happens, but we know that it occurs in specific cells where we have high levels of certain gene products are necessary. Now, if you think about it, in the liver is responsible for cleaning a lot of things out of our system. And so there may be times when you have to have lots and lots of one particular type of product that's produced. So if you have more of these genes available, so if you have more chromosomes available, you have more product, or you have more potential product available as well. So this is definitely a good thing, especially within the liver itself. All right, our next topic is going to be rearrangements of chromosomes. We're actually going to look at these individually, but we're going to look at deletions, duplications, inversions, non-reciprocal translocations, and reciprocal translocations. So before we go any further, I'm pretty sure you understand what deletions, duplications, maybe not so much inversions means, but definitely deletions and duplications. But let's define what translocation is. So a translocation is an exchange and transfer of pieces of chromosomes. So this is going to be segments of a chromosome that are actually going to be exchanged or transferred between individual chromosomes. So this is just a general idea of what we're looking at. We are going to look at each one of these individually, so that you can look at and see how these things are actually occurring. Maybe we are, if I can advance the slide. There we go. All right, so deletion of D. So what's happening here is you have a complete chromosome, and I'm sorry that you can't, well, I hope you can see all of it down there, but if you can't, I apologize. So you have, for this particular chromosome, you have, I'm going to switch to a different color here. You have genes A, B, C, D, E, F, and G. Now something happens in this process, and as you move from, or as you come from this particular chromosome, which is our normal chromosome, okay, what happens is, notice that you now have A, B, C, E, F, and G. So we've had a deletion of this particular segment, somewhere around in here. We've had a deletion of that segment of the DNA. Now notice that so this dark area here, this would be your gene locus. So that is where gene A is sitting. So notice that in order to achieve a jump from C to E over here, we've actually lost a little bit of this region here and this region here, such that you have the same distance between C and E. This doesn't always happen, but in this particular cartoon, if you will, this is what's going on here. So you've lost that particular piece of the chromosome. And we're going to talk about ways in which these kinds of things occur later on. Now this is a duplication, and so that's sometimes a little bit easier to see because you have, you're starting out with genes A, B, C, D, E, F, and G, just like we had on our original chromosome before. But now look what happens. We have A, B, C, B, C, D, E, F, and G. So here what has happened is, so we have a normal representation here, but this section is actually duplicated. Now you might not think that this happens very often, but actually sometimes when we are actually copying the chromosome, polymerase gets confused. And sometimes polymerase, as it's actually moving, and this is not an exact representation, obviously, because we're looking at a chromosome and not unwound DNA. But if you have DNA that's uncoiled, then what happens is polymerase sometimes skips. So it can skip, or it can be confused and actually back up and copy the same piece again. Now we hope this doesn't happen very often. Okay, so if this does happen, you now have a chromosome that has 2 extra genes on it. So for this particular individual, we would say that if everything else goes all right, if this chromosome is the one that makes it into the gamete, so if this chromosome is transmitted, then what you're going to see is that you're going to end up with an individual that is trisomic. Well, I don't know what happened there. And I should probably stop doing that, whatever it is. So let me erase this. So it's going to be trisomic for B and C here. I think my pen went away. There we go. For B and C. Okay? So if an individual receives this chromosome along with a normal chromosome, then you will have 3 copies of B and you will have 3 copies of C. And we talked about that this isn't always a good thing because you can't compensate for the dosage in this particular sense. So this is an inversion of B and C. And so what happens is you actually have a situation where in our inversion, so you have A, B, C, D, E, F, and G. So again, this is our normal chromosome, okay? But what happens in an inversion is that you have A, which is in the right spot, but D is now not in the right spot, C is in the right spot, but B is not in the right spot, and then everybody else is still okay. So here's the problem. So if we have A, D, C, B, E, F, and G, how is this matching up with our normal chromosome? And the truth is that sometimes it cannot match. If this is the case, if it cannot match, then sometimes you will not be able to actually form viable gametes from this particular individual. So this is an inversion of B, C, and D. And the next thing that we're going to talk about is some sort of translocation. Okay, so there are 2 different types of translocations. We are actually going to talk about non-reciprocal, so non meaning not, so non-reciprocal translocation. And if you remember, we have 2 different chromosomes here. So remember, our colors mean something different. So this we're going to call chromosome 1. And this is going to be chromosome 2. Okay? So on chromosome 1, we have our normal chromosome that we've been working with before. A, B, C, D, E, F, and G genes. But notice that this is chromosome 2, and these are not homologs, correct? Because we don't have the same genetic complement. So they are non-homologous. And here we have H, I, J, K, L, and M. And in this sense, what you have here is 2 different chromosomes that are actually going to exchange DNA with each other. Now we know that this is not the best option, and we know that it shouldn't happen very often. But what has happened in this particular instance is our chromosome 1, A and B, has now actually been translocated on top of, remember this is chromosome 2. Okay? So what happened to chromosome 1, and it's the reason that it's non-reciprocal, is because we didn't have the same amount of transfer. So we lost A and B here, right? And we gained A and B here. So now what's happened is my chromosome A here, or chromosome 1, sorry, now cannot match up, probably can't find the homolog over here, because these two are now very different from each other. Okay? So they could match up, but only here. Okay? So we will not have the ability to actually match this section in this particular chromosome now. Okay? So what happens here in our second chromosome is that we have gained a portion. So we gained A and B, but this doesn't really necessarily mean that we can't match this section, with this entire chromosome, because it's still actually pretty well lined up, or it can line up. Okay? But this would be a non-reciprocal translocation because we don't have the same amount of same amount of genetic transfer between chromosomes. This is reciprocal translocation, which means by definition, because we said a non-reciprocal translocation means that we don't have the same amount of DNA that's actually being exchanged, that means that a reciprocal translocation does have the same amount. Okay? So we're starting with the exact same complement that we had before. So this is chromosome 1, and this is chromosome 2. And if you'll notice, basically what's happening here is we have the same piece of chromosome 1 that's transferred to the second chromosome, and we have a chromosome set that's actually been transferred here. So while it's not the same amount of DNA that's actually being transferred, a reciprocal translocation just means that we are changing DNA between two chromosomes. So if memory serves, I said that it had to be the same amount of DNA, and that is not true. It's the fact that we are transferring DNA from one chromosome to the other, and then there's also a reciprocal translocation of DNA from the other chromosome back to chromosome 1. So this would be a reciprocal translocation because these two pieces are actually being transferred. So is it your opinion that we have the same genetic complement by looking at these two chromosomes? And the answer is yes. So if an individual inherited this chromosome and this chromosome, they would actually have the same genetic complement because they do have A, B, C, D, E, F, and G. So that's the same thing as over here, A, B, C, D, E, F, and G. And they also have H, I, J, K, L, and M. Same thing over here, H, I, J, K, L, and M. So they have the same genes, as in the normal chromosomes. They just aren't carried now on the same chromosome. Now this still does present a problem, because can these two chromosomes match up with each other? So if we look at these chromosomes together, then they can't match up with each other. Okay? So these chromosomes here will not be able to line up with each other because the centromeres can't find each other and the DNA can't find each other. They still may be able to minimally actually, so A and B here could line up with A and B here, but that's it. H, I, and J could line up here, but not so much with anything else. So it's still going to be a problem if you have two individuals that are, if you have a normal complement trying to match up with this, which would be an abnormal complement at this point. All right, so let's talk about deletions. Deletions are missing chromosomes, and so our missing chromosome is basically when we either have a chromosome that breaks in one or more places, and then a terminal portion, and a portion is lost, but the portion can actually be either terminal or can actually be in the middle. So if a portion is lost, the missing piece is referred to as deletion. Sometimes you will actually see it called a deficiency as well. And the book kind of moves back and forth between these two terms. So you'll see deletion, but you'll also see deficiency, which is why I'm presenting both of those. The deletion can occur near one end. So this is referred to as a terminal deletion. But there are some instances where we can have losses from the middle of, or not necessarily the middle, but the interior part of the chromosome as well. This is referred to as an intercalary deletion. The portion of the chromosome containing the centromere region will usually be retained because we have to have that centromere that's going to hold things together. So usually what happens is the deletion is outside of the area that involves the the centromere. So let's look at these. So basically what we have in this particular, the first part of the diagram up here. So let's look at just the top portion here. So this is, stop doing that. No. Okay, I don't know what happened there. I don't want to extend my page, I want it to go down. Okay, so what we have is, let me go back one, and I'll go back to what we had, and it didn't work that way, did it? Okay, stop doing that. New technology. Okay, so the origin of the terminal deletion in this particular one here, so this is A. So with A, notice that we go from A, B, C, D, E, and F, and we've lost A. So this whole section is now gone. So remember, if you'll remember back to what we talked about with Patau syndrome, Patau syndrome was actually a, Cri-du-Chat. Wrong one. We did talk about Patau as well. But when we talked about Cri-du-Chat, what we were talking about is that this particular one is a loss. So it's a monosomic event, and so it's a small part of the short arm of chromosome 5 that's actually missing. And so this would be an instance where this is actually how you could have Cri-du-Chat that actually forms. So something happens, there's probably a weak spot here in the chromosome, and you have a break when these two chromosomes find their homologue and try to pull apart. You can actually lose a piece in disjunction. Okay? Now, another way that this can actually happen is an intercalary deletion here. And so basically what happens is you have a malformation of how your chromosome is actually lining up. So notice that we have a loop that's actually formed here. Okay? Now, this is not normal, and we don't want this to happen. But if your chromosome is not linear, if it has become in some way entwined with itself, you'll actually have this type of twisting that does occur. So what happens if that does happen, if you have crossing over between these 2 points, basically what can happen is you can lose a section of your DNA here. so your D and your C. And then what happens is these two parts of the chromosome will actually come back together again, so that you now have a chromosome that has a deletion, and D and C here are actually just floating out there in the cytoplasm. So they're not going to become anything that interferes with anything else. They're just going to be lost. And so this will not then move forward to the next generation. Or if it is, it will not be active. Okay, so it's not active if it's out there by itself. So the last one that I'm going to try to pull the slide back up so that you can see this. Okay, and now I have to erase. Is the formation of a deficiency loop. Okay, so a deficiency loop is just as suggested. It is an area where you have a missing portion of a chromosome. And so here is your, if you'll refer back to our deleted chromosome that's above here. So our deleted chromosome is now down here. So we now have a normal chromosome that is trying to pair with a abnormal chromosome, so one that contains a deletion. So the only way that this can actually happen is if A and B match up with each other, E and F. Remember, these are genes we're talking about here, genes that are carried on our chromosomes. And there's actually nowhere for C or D to actually match up here. So what happens is you have A and B that are matching up, and then you have, would all be homologous DNA as well, and so would this. And notice that your centromeres have found each other too. So there's nothing here for this particular part of your chromosome to match up with. So it forms a deletion loop. So it's a loop in your normal chromosome that allows it to match up with your chromosome that has a deletion. So what can happen is you can actually have an individual that inherits a chromosome that has a deletion and also has a normal chromosome, and can still then pass this, if it matches nicely, can still pass this to the next generation. Okay? So it can actually be passed to the next generation, and then this can actually be perpetuated in future generations. So a duplication can actually occur as well. So just as we have deletions, we can also have duplications as well. A duplication actually arises as the result of unequal crossing over. And we've talked about crossing over in terms of the ability to reproduce a wild type if your mutations are further apart. But now remember, we are talking about chromosomes. So now we're talking about crossing over between two chromosomes. And you can actually have an unequal crossing over that occurs during meiosis, or through a replication error that actually occurs prior to meiosis. So again, remember that we can have a duplication that occurs if we have polymerase that skips and then backs up and then copies the same set of genes again. So you can actually have two or more genes that are actually copied through that duplication error as well. But what we're going to talk about now is a deletion that actually, or excuse me, a duplication that arises as a result of an unequal crossing over. So this is what we're dealing with here. Okay, so in this particular situation, we have two sets of. I'm not sure why this keeps doing this, but it bothers me. All right, so we have our chromosomes. They are actually, so you'll notice that these are homologs. Okay, so we have, in this particular instance, we have these two individuals that are homologs and these two individuals that are homologs. They are different colors, so that means they're not exactly the same thing. So this is going to be, notice that they have the same genes, so this would be, these are sister chromatids. They are homologs, but they are sister chromatids. And they are duplicated chromosomes. So remember, these are belonging together because we have centromeres holding them together. So they are sister chromatids, and these two individuals, so this is chromosome 1, and then we also have a set here that would also be chromosome 1. Okay, so what we have here are is a crossing over event that's actually occurring between these two. Now, it's unequal crossing over because what has happened in our alignment? So these two, if we're looking at this, our centromeres should actually be lining up with each other, and they are not. So what's happening is that our genes are not lining up with each other either. Okay? So what happens in this particular instance, we have an offset chromosome arrangement, and we have crossing over between two non-sister chromatids, right? So this is how it's supposed to be, but then what's going to happen is we're going to have, when these separate, we're going to have one normal chromosome, right? So that's indicated here. Let me get back over here. Okay, so we have one normal chromosome, and that is going to be reflected in the one normal chromosome here, right? So A, B, C, D, E, and F on this particular chromosome, and A, B, C, D, E, and F on this particular chromosome. And then notice that what we're going to have, so we're finished with this one, so let's look at the next one. I'm going to erase some of this stuff. It's getting too busy. Okay, and then what's going to happen is, if we look at this, we have this particular chromosome, so 2 is now doing this. Okay, so now what we have is, over here for chromosome 2, we now have A, B, and then it's going to cross here, D, E, and F. Okay, so now what we have is an abnormal chromosome that only has A, B, D, E, and F. Okay, so let's get rid of this one. And then what's going to happen is, if we're looking at 3 here, if we start with 3, we're going to go A, B, C. Uh-oh, we picked up another C, D, E, and F. So for 3 over here, we now have an abnormal chromosome, and we have a duplication of C. So we have A, B, C, C, D, E, and F. So if we now look at our other chromosome over here, we end up with A, B, C, D, E, and F, which is normal. So if you look at chromosome 4 here, we now have A, B, C, D, E, and F. So our 2 normal chromosomes are actually going to be 1, 1, and 4. But 2 and 3 are abnormal. So we have a deletion. So we have a loss of C on 2, and we have a duplication of C on 3. So those are abnormal chromosomes. So this is actually how we have this particular or these types of mutations that actually occur. So this is a duplication and actually a deletion as a result of crossing over error. So one of the other things that we need to talk about is gene redundancy and amplification. So ribosomal RNA has to be in abundance to support protein synthesis. And in order for this to occur, we have to have lots of different copies of things. You wouldn't want one copy of an ATPase gene because you have to have lots of copies of ATP. So if you only had one copy of an ATPase gene, you'd have to wait a whole long time to accomplish a lot of the work that's necessary by having multiple types of and multiple copies of ATP out there. So organisms have multiple copies of our ribosomal RNA genes. This is called gene redundancy. It helps because if you have one problem with one particular gene, then you can actually make this work with other genes. But it also actually allows you to increase the amplification of our DNA or our RNA. Okay, so basically what happens is you have a normal redundance of rDNA, so these are your ribosomal DNA genes, is insufficient to provide adequate amounts of rRNA and ribosomes. So now you have to have lots of different copies. It means you can make copies of all of these at the same time or at different times. So this gives you lots of transcripts that are necessary for development following fertilization or for other processes that are actually necessary. So if you'll notice that that in some of the oocytes of Xenopus, which is one of our standard organisms that we use to look at development, so this would be a model organism for development, has about 400 copies of some of these genes in the cell because they need that many copies in order to make sure that there are enough transcripts out there to help with development, because you need a lot of copies of those things when you're trying to divide as many times as they have to. Now, in the same idea, we can actually produce new genes through gene redundancy and amplification. So if we have gene duplication, so if you look back at that chromosome that we ended up with 2 copies of C on, maybe we don't need 2 copies of C working on that particular chromosome. So maybe one has an opportunity to evolve into something else. So if a gene is duplicated, one copy will actually still usually perform essential functions and allow the organism to live, while the second copy is either turned off or is allowed to accumulate mutations. And the product of that particular gene, so the one that it's accruing mutations, can actually acquire a new function. And so we can end up with what we call gene families because you get a new function in the second copy that may actually increase the fitness of the organism. So as a result of this, gene duplication actually may play a role in evolution. And we do have support for this particular hypothesis by the discovery of genes that have a substantial amount of their DNA that's in common. So you have 2 different genes, and they are almost identical to each other if you sequence them, but their product is not the same. So that means that the protein or the enzyme that they are producing is going to be distinct. So it's not going to have exactly the same function. Other support for this includes the presence of gene families. So this is what I alluded to before. So we can have gene families, and basically what a family is a regional group of genes whose product performs the same general function, but is a slightly different form of the gene. So we do know that we have globin genes that are or part of a gene family. So a globin chain is part of a hemoglobin. So we need globin, we need alpha globin, we need beta globin, and several others that can actually be formed together to actually make up hemoglobin. We also have lots of different types of T cell receptors and antigens that are out there. And a lot of our major histocompatibility complex is the reason that we have such a variation in these genes is because we have different genes that are forming more and different types of proteins that are actually found on the outside of our cells. So these are genes that are found in all species, and they are typically highly conserved genes because they are performing a specific function. You wouldn't want all of your globin genes to be so different across species, because then they wouldn't have the same function. So, but we do know that through time that we have evolved different types of these genes that actually work together to perform a, well, they have different products that are working together for a specific function. Okay, at this point, we're getting close to the end of our time. About 20 minutes left. We're going to be talking about inversions, where we are looking at rearrangements of the linear gene segment rather than a loss of genetic information. So we're not talking about necessarily duplications, and we're not talking about deletions. In this particular instance, we are talking about just rearranging the order of the genes. Now, as we have talked about before, some genes have to be next to each other in order to perform a specific function. If this is the case, if you reorder the genes, then you may not have the same function. In this particular instance, an inversion would involve a segment of a chromosome that's actually turned around about 180 degrees within the chromosome. Now, this is a difficult one because it requires two breaks in the chromosome, one above the section that's going to be inverted and the one below the section that's going to be inverted. And then you have to have a reinsertion of that inverted segment. This means that you have to have two breaks in your chromosome. This chromosome then has to flip and then has to the ligase actually has to reattach these segments back to each other. Now, we do know that this can actually occur through a process called chromosomal looping. So let's look at an example of this. So this is actually going to be chromosomal looping. So what happens is you have a normal chromosome. It is linear. And for some reason, this chromosome now actually loops back on itself. Again, this is a process that could happen through a lot of different ways or a lot of different mechanisms, but essentially what happens is you actually have your, maybe it's tangled, something's happening, and so you have a chromosome that's now looped back on itself, okay? Now if you have, so now that we have the loop here, if we have a break at this point, what happens is you now have this piece, you have this piece that's separate, and you have this piece, okay? Which then creates this particular setup over here. So that we now have E and F that are apart from each other. So these should be connected, they are not, and these should be connected and they are not, okay? If we actually have a situation where these are put back together again, this is what happens. So you're going to get A, E, D, C, B, and F. So in this particular instance, we now produce a chromosome with an inverted sequence down here. So we now have A, E, D, C, B, F. So if you tried to match this chromosome with this chromosome up here, obviously there's going to be differences. And it would be very difficult for these chromosomes to match up with each other if they are now found in the same individual and are acting as homologs for each other. Another type of inversion that actually does occur is called a, well, let me make it forward. There we go. A paracentric inversion. We have paracentric and we have pericentric. We're going to talk about these separately. So we have a paracentric inversion, which does not change the relative length of the two arms. So now what we're talking about here is an arm A. So this is what arm? Hopefully everybody remembers that this is our petite arm, our P arm, right? And then this is our Q arm. Okay? And so what happens is, what this is showing you is that we have a break at this point right here, and we have a break at this point right here. Okay? Now we end up with the same number of genes, A, B, but instead of C, D, E, and F, we now have E, D, C, and F. So this is still an inversion. So we still have a gene inversion. But we refer to this as paracentric because it doesn't have a change in the arm length. Okay? Now we also then have a pericentric inversion, and the pericentric inversion does have a difference in the, or does have an effect on the distance in the arm ratio. Okay? So, in the length ratio. So what happens here is we have A, B, C, D, E, and F. So that's our normal chromosome from above. But now what happens is we have A, D, C, B, E, F. So the section that was inverted was this section here. So I'm going to choose a different color. We had an inversion between B, C, and D. So this is now D, C, and B, and has changed the ratio of the arms such that we now have a total of 3 genes, and on this side of our arm, this side of our centromere, and 3 genes on this side of our centromere. So we now have our centromere goes from, in this particular instance, well actually, sorry, pointed to the wrong one. So if we look at our 2 chromosomes here, we now have a metacentric arm, well, ratio, because our centromere is now metacentric. Okay? So we have a metacentric centromere. Okay? Because it's now occurring in the middle of our chromosome, and it wasn't doing that before. Okay? Now, although we only have a couple of minutes left, I want you to actually look back through some of these slides that I'm going to go through next, because they are the most difficult for people to follow, and I want you to understand what you're looking at here. Okay? So the next one we're going to talk about is, and this is something you may want to review a couple of times, we're going to talk about synapses of inverted chromosomes. That means inverted chromosomes are ones that have an inverted or a changed order of genes on those chromosomes. And we're going to actually, as a result of this, require an inversion loop. Okay? So if you remember what that is, remember that your chromosome that's trying to match with one that has an inversion actually forms a loop to be able to move on and change. So what we have here, and this is a paracentric inversion, so hopefully you remember what that means. We do have an inversion of, so normally we have A, B, C, D, E, and F. And here we have A, D, C, B, E, and F. So our inversion is here. Okay? D, C, and B. Okay? Now, we're going to move to these 2 sets. So this is chromosome, we'll say this is chromosome 12. And this is chromosome 12. We can say this is from dad, and this is from mom. Right? Okay? So what happens is these are trying to actually match up and find homologs. Okay? So notice that you're going to pair not across all 4, right? Okay? But you're going to have pairing between the two sister chromatids here. Okay, so now if we move down below and we actually look at what's going on here, if you move across here, you're going to go 1, well, we're going to start with 1. Okay, we're going to go A, B, C, D, E, and F. So notice this notation here. 1 starts here, and 1 prime starts or ends here. Okay, so that's the notation. So that means that this chromosome 1 to 1 prime is a normal sequence, and there's nothing changed. Okay, because remember, this was not our, so this chromosome up here was not inverted. It is normal. Okay, now let's look at what happens by looking at our other chromosomes here. So we've already done this one. Now we're going to look at what happens with 2. We start with 2. We have A, B, C, D, E, and F going out to 2 prime. Well, we then again produced a normal sequence. So our 2 to 2 prime is also going to be a normal sequence. Okay? So we didn't do anything here but just match up. Okay? Now the one that has to change is the fact that this one is inverted. Okay? And so let's look and see what happens when we start looking at our inverted chromosome and how it's having to match up. Okay? So now what happens is we end up with, starting with 3, A, where does it go? It actually goes D, C, B, E, and F. So if we go 3, 3 is going to lead us to 3 prime, but we're going to have still our inversion, right? D, C, and B. Okay? So these chromosomes have lined up, and they can still maintain the inversion, and we're going to end up with a chromosome with an inverted sequence of D, C, and B. Okay? Now, what happens when we look at, let me pull this up a little bit. Okay? So now let's look at what happens with 4 here. So we're going to start with 4. And 4 is actually going to go A, D, C, B, E, and F. So we still maintained, when we start with 4 here, and we go to 4 prime, we still maintained the exact same sequence, right, in our inverted chromosome. So we didn't do anything different here, okay? We're still maintaining our inverted sequences on these 2, and we are still maintaining a normal sequence on these 2. So this individual, the resulting gametes, are still going to produce the same complement. So are these capable of producing a viable individual? So the answer is actually probably yes. Because they have all of the genes that they need. So none have been lost, none have been added, none have been duplicated, but they are in a different order. So as long as there isn't a, so if we say that there is a need for a specific order, they will be okay. So we will be able to have individuals that can live a productive life, but they may not be able to, potential that they may not be able to produce or reproduce viable young with this particular crossing over type. Well, not crossing over, but matching type. Excuse me. So the last thing that we're going to talk about is actually a crossing over event that is an inversion loop with a paracentric and pericentric inversion for heterozygotes. So basically what happens here is a crossing over event. So what we talked about before was synapse, so it was not crossing over. So basically what's going to happen is we're going to have one recombinant chromatid that's dicentric. It will actually have 2 centromeres, and one that will be acentric or will lack a centromere. So let's look at this example here. Okay, so this is a paracentric inversion heterozygote. Okay, so what happens is, and this arrow down here is actually supposed to pop up later, but we'll deal with that. So paracentric inversion, what we have is a heterozygote. So we have heterozygotes for both alleles, meaning that we have a dominant copy and a recessive copy for each of our alleles. So this is a heterozygote. Notice that this is now an inversion loop that includes crossing over. Okay, which means that our chromosomes are going to exchange DNA, okay, between non-sister chromatids. Okay, now what happens here is if we start with 1 and we move to 1 prime, just as we did in the previous diagram, we're going to go A, B, C, D, E, and F all the way to 1 prime. So what happens is we're actually going to have a normal sequence that is produced for this particular chromosome. So nothing is different about this one. We didn't cross over. So no crossing over, normal sequence. Okay? So now let's look at what happens, and notice that this is where crossing over is actually going to occur, because this is where our 2 non-sister chromatids have actually attached to each other. So now what we have happening is we start with 2 and we move A, B, C, now across here, D, A, and so what we end up with is a 2 to 4. So not a prime, so we didn't end up with an end over here, right? So we went back to the front of a chromosome. We started at the front here, and we went back to the front here because of crossing over. So that means that this individual now has 2 centromeres. So this is a crossing over and dicentric duplication. So notice that we have 2 A's and a deletion of E and F. Okay. All right, so let's look at the next one. Erase this, it's a little messy. Okay, so if we start with 3, we're actually going to go through our centromere A, around, and back out to 3 prime. And what we see with this one is that if we start with 3, we do have an inversion. So we have DCB here instead of ABCD, but we have a non-crossing over inverted sequence. So this is the same as what we saw up here. Okay, a non-crossing over inverted sequence. All right, so our last one is, let me pull this up a little bit. So let me pull that off. And let's pull up. Oops, I forgot about him. Okay, so now what we have is, let's start with 4 here. And we start with A. But now this time we're going to go this way. And we have to go back up and catch this. So, oh, wait a minute. I did the wrong thing. Sorry. Don't pay attention to that. Don't be looking at it. We already did 4, remember? Or I didn't. So remember, we already started out with 4 here. Okay? So because we already started out with 4, okay, I can't start there. So I've already completed chromosomes that end with, or that start with 1 and 2 and 3 end with 1 prime, 4 prime, or 4 and 3 prime. So that means that my end that I haven't actually worked with yet is 4 prime. Okay? So this time I'm doing this. I'm starting with F, E, and that's my time. Okay, but we're going to finish this one. So 4 prime, we're going to start with E, F, B, C, D, E, and F, and actually we're going to come out with 2 prime. So remember that with this one we started out at a beginning and we ended with a beginning. Now we're starting out with an end and ending with another end. So what happened to this one? We have no centromere. So we have here, we have crossing over. It's acentric, so we don't have a centromere. And we have a duplication of E and F and a loss of A. A's missing. Okay, so we have a deletion and we have duplications and we have a loss of a centromere. So you can imagine that this is going to actually be a problem for this particular chromosome to find anybody to line up with, because there is no centromere for this one to actually match up. So we will have no matching between this particular chromosome here and a homologue that may actually occur. Okay? So we'll end here. So I tried to manage it as a 75-minute session, just like we would have in class. Please do watch this and be ready to ask questions in class. I'm not going to reteach the entire lecture, but I am definitely going to address any questions that you may have with any one of these. And so hopefully the opportunity that you have to watch this a couple of different times will help you understand the information a little bit more, because this is a little bit more of a difficult lecture to be able to follow and to see what's going on here. So please remember that all of this material is available for a quiz that may occur on Monday, or, well, actually probably Wednesday in class. Okay, so with that we will stop.